For over a century, the scientific community has been racing to decode exactly how the malaria parasite evades the human immune system. The most lethal of these parasites, Plasmodium falciparum, survives by constantly shifting its appearance. It relies on a hypervariable family of genes called VAR genes, which dictate the specific proteins displayed on the surface of infected red blood cells. By switching which VAR gene is active, the parasite stays one step ahead of our defenses. To study which VAR genes actually drive severe disease, researchers must extract the parasite's RNA directly from a patient's blood. The logistical problem is that clinical blood draws often contain very low parasite counts, yielding far too little RNA for comprehensive sequencing. The standard operational solution, employed by top research institutes globally, is to take that clinical isolate and place it into a petri dish. By letting the parasite replicate in a short-term in vitro culture, usually for 1 to 3 cycles, researchers can harvest enough biological material to run their sequencing pipelines. This workflow rests on a single biological assumption, that keeping the parasite in a culture dish for just a few days perfectly preserves its original in vivo phenotypic and transcriptomic state. Decades of data regarding disease severity, patterns of drug resistance, and host interaction dynamics are predicated entirely on this assumption holding true. If moving the parasite from the human bloodstream into an artificial environment alters its VAR expression, our primary window into severe malaria pathogenesis is compromised. Testing whether this assumption holds true has historically been incredibly difficult. Older analytical methods, like quantitative PCR or DBL alpha tagging, simply couldn't capture the full complexity of the VAR gene family. Even modern RNA sequencing struggled. Because VAR genes are incredibly polymorphic and mutually exclusive, they cannot be accurately mapped against a standard, static reference genome. The sequences simply do not match up. Researchers recently developed a novel computational pipeline to bypass this roadblock. Instead of mapping against a reference, this pipeline uses a de novo whole transcript approach to assemble these hypervariable genes from scratch, directly from the raw sequencing reads. When this pipeline was applied to paired samples, comparing the exact parasite isolate drawn directly from a patient to its first generation cultured in the lab, the transcriptomes did not match. Within a single replication cycle outside the human host, the parasite undergoes massive, unpredictable transcriptomic drift. Placing a sample into culture triggers an aggressive biological perturbation, overwriting the exact clinical signatures researchers are attempting to study. To see how the new pipeline captures this drift, we have to look at how it isolates the data. First, the algorithm takes the raw RNA sequencing reads and filters out any genetic material belonging to the human host. Next, it maps the remaining data against the standard 3D7 Plasmodium baseline genome. Any read that matches this standard core genome is discarded. What is left is a small, highly concentrated pool of non-core genetic data. These isolated reads are then fed into the RNA spades algorithm. Instead of trying to match fragments to an existing map, the algorithm performs true de novo assembly, weaving them together into long, contiguous chains based solely on overlapping sequences. This whole transcript approach successfully reconstructs the complex architecture of the VAR genes. Specifically, it pieces together the highly variable N-terminal segment and the DBL alpha domains, the exact structures that dictate how the parasite binds to human cells. Older bioinformatic pipelines frequently failed at this stage, producing fragmented or internally misassembled sequences that scrambled the parasite's true genetic profile. By abandoning the reference genome and building from the ground up, researchers finally have a high-fidelity tool capable of observing exactly how the VAR transcriptome shifts in real time. Equipped with this tool, researchers conducted a paired analysis. They mapped the VAR expression of a patient's isolate the moment it left the body and compared it against that exact same isolate after 1 or 2 replication cycles. When we plot these results on a principal component analysis graph, the initial ex vivo data points cluster neatly by patient identity. But look at what happens in culture. The Generation 1 and Generation 2 points vector sharply away from their ex vivo origins. While some samples maintain a dominant clonal expression profile in the first generation, others exhibit immediate, erratic VAR gene switching. The laboratory environment induces sudden shifts in which surface proteins the parasite chooses to express. By the second replication cycle, a biological convergence point emerges. The parasites begin systematically abandoning their clinical, patient-specific VAR profiles. This line graph tracks multiple patient samples, showing a uniform upward trajectory in VAR2 CSA expression. When subjected to environmental stress, the parasite uses this specific gene as a default pathway, a transcriptional sync node that it reverts to when survival parameters suddenly change. The VAR repertoire is the exact mechanism we study to understand disease severity, and yet this data shows the laboratory environment artificially overwrites it in a matter of hours. If the hypervariable genes are switching, we have to look at what is happening to the baseline, non-polymorphic core biology of the parasite. This volcano plot illustrates the scale of the systemic shock. Upon entering culture, 19% of the parasite's core transcriptome, over 900 individual genes, is significantly differentially expressed in the very first generation. This spike is driven by massive upregulation in specific functional groups. We see an immediate flood of stress response proteins, RNA-associated proteins, and AP2 transcription factors that are explicitly linked to how the parasite invades host cells. Looking at a new PCA plot for core genes, we see how this stress response collapses the clinical heterogeneity of samples. Ex vivo samples map out widely based on the patient's condition, but Generation 1 samples collapse into a tight, indistinguishable cluster. Distinct clinical signatures are wiped out entirely. Ex vivo samples show clear, measurable transcriptomic differences between a patient who is naive to malaria and one who has been previously exposed. By Generation 1, that distinction completely vanishes. The culture dish effectively sanitizes the parasite. It strips away its unique host environment history and replaces it with a generic, lab-induced stress profile. The human bloodstream is an intensely hostile environment. The parasite faces immense immune pressure and tightly restricted nutrients. In contrast, the laboratory culture media is an artificial paradise, an immune void with abundant resources. Removing host immune pressure and flooding the system with generic nutrients delivers a severe environmental shock that forces rapid adaptation. Under a microscope, this deception is visually complete. The parasites appear to be thriving, replicating perfectly normally inside the cultured red blood cells. But this physical survival masks a transcriptomic illusion. At a genetic level, the organism on the slide is no longer the organism that caused the disease in the patient. These findings require that any inferences linking VAR expression to severe malaria phenotypes be drawn exclusively from raw, uncultured ex vivo samples. Achieving this requires the widespread adoption of advanced de novo assembly pipelines. We must process low parasitemia clinical data directly without relying on the crutch of in vitro expansion. To decode the pathogenesis of Plasmodium falciparum, we must abandon the in vitro illusion. We must accept the complexity of the raw data and meet the parasite exclusively on the battleground of the human host.